This is me here, okay? And this is going to be sort of a technique kind of talk here. So it's not heavy on a ton of data. You'll be glad to hear that. And we're going to talk about some things that some of you know about already, but it might be new to some of you, which is how to develop your assays a little more reliably from the coupling step to making your bead mixes and stuff like that. So some of the things I'll go over, which some of the other presentations actually went over better than this in their slides than I'm going to, but I'm going to be quicker here. A little quick review of coupling chemistries, then some of the resources and tools that can be used for developing assays. And then I'm going to talk about this molar versus microgram coupling, because you saw in a lot of the talks today that a lot of people were coupling their proteins or whatever at sort of micrograms per million bead kind of thing. But I'm going to show you some of the advantages of doing molar coupling in some applications. And then I'm going to show you the tools for performing these kinds of couplings. And then even though doing the coupling is an important part of developing your assay, there's also the ability to make your bead mixes properly, to make sure your assay can work properly. So these are the topics we're going to cover. So first, coupling chemistries. Pretty much everything on the Luminex platform is dependent on the carboxyl group there, where we pretty much activate this carboxyl group with EDC so that something that's got an amine on it can couple to the beads covalently and not go anywhere after that. So because of that, things you can couple are going to be various proteins and antigens. Of course, a lot of people are doing antibodies. There are some peptides you can couple. Once in a while, if you got a carbohydrate with an amine group, you can couple them as well. A number of small molecules that have amines on them can be coupled as well. And if you want to develop a genomic assay and you've got an amine modified oligo, you can couple those to the beads with these activated carboxyl groups as well to develop a genomic type assay. Now, for some smaller molecules, like things like peptides and whatnot, you may need to put a linker on the bead. And these can be various protein linkers, such as BSA, KLH, thyroglobulin, or avidin. And we actually sell some avidin-coated beads because in this scenario, you might have a biotin tag molecule. It could be a peptide or something else that you want to bind to the avidin beads and not go through that whole process of doing the coupling, which takes a few hours, as I'll show you shortly. And then there's also some small molecule linkers you can add as well, coupled to the beads with the same chemistry for covalently linking other types of proteins and stuff to the beads as well. So if you're ever interested in talking about any of these, get in touch with either tech support or one of us field application scientists, which in the US is me, and we have another FAS, Gabby. Are you here, Gabby, somewhere? Yeah, she's way in there in the back, hiding in front of the thing back there. But you can contact one of us to figure out what you need to do for your assay to improve it. So for protein chemistry, if you got a couple of protein to the bead, coupling something large, say like this is an IgA molecule, is pretty easy because it has a lot of lysine groups that will react with the activated carboxyl groups. Some smaller proteins like various antigens and whatnot, they'll have fewer amines on them, but lots of times you can still couple them fairly effectively. When you start getting into things like peptides, sometimes they may have no amines on them or maybe only the terminal amine, or they may have a few amines in there that will allow you to couple them. But still sometimes these are so small you might not want to couple them directly to the bead. And we have a couple of other ways that you can do that, which I won't go into here because you guys all want to get to the happy hour, okay? So some of the resources and tools for developing any assays on the Luminex platform, the best one to start with if you're not sure what to do, which I think most of you do at this point, but the cookbook is a good place to go to get sort of easy to follow initial protocols with all the kinds of reagents you'll need to use, list of equipment, to develop different types of assays on the xMAP bead technology. But I think most of you have gone through this already, and you may be wanting to contact us for maybe some tips and tricks about some of the problems you might be having. But if you have anybody that wants to start, tell them to download the cookbook. So again, that carboxyl group is the key to getting it activated to couple anything to the bead. So for proteomic assays, the workflow is pretty much something like this, which one of the other speakers had a nicer slide that I might have to borrow from him in the future, But it summarizes the same thing. You have, the first thing is you take your beads and you put them in the tube. You wash them to get out some of these preservatives in the tube out of the beads, and you can usually do it with water or activation buffer. Then you'll resuspend it in activation buffer, put in your sulfur NHS and then EDC, incubate that for 20 minutes so that you can activate those carboxyl groups. Then you'll wash the beads to get out, the EDC and sulfur NHS, and then you'll add your desired amount of protein per million beads, incubate that for up to 2 hours, and then do a few more washes. And for most protein assays, you can use our PBS-TBN buffer, which has BSA in there to block any active carboxyl groups that might be remaining. Now, there are a few situations where you might not want to use this buffer with BSA, depending on the size of the molecule you're coupling. But again, it's the kind of thing that if you're having some problems, give us a call and we'll look at your protocol and decide if there's something else you need to do at this step that's different. So now let's talk about some of the microgram versus the molar coupling. Because a few years back, I was starting, let me backtrack a bit. Because in the cookbook, the cookbook recommends, using 25 micrograms of protein per 5 million beads. So this works out to be 5 micrograms of protein per million beads. But as users, most of you have probably tried, more or less than this if you're coupling an antibody or an antigen to try to optimize your assay for the best performance you can get out of the assay. So for smaller proteins like the peptides, though, you might need that molecular linker of some type, whether it's another protein or another, smaller linker molecule. And you can still couple those at micrograms per million if you want to get these other molecules onto the beads. But there are some situations where you're coupling proteins, and you might want to do it at moles of protein per million beads. And this can be better when it's advantageous to have sort of the same number of molecules on the bead surface if you're coupling proteins of different molecular weights. Because what happens is when you're doing these molar amounts per million beads, this allows you to sort of control the number of molecules you have per bead so that the signal is going to be dependent on the specificity of something reacting with those antigens and not dependent or reflective of the number of molecules on the bead. And you'll see how that's important in a second here. So this type of approach is really good for a number of serological assays where you're looking, say, for an antibody titer or you're testing antibodies for their specificity to a given antigen. So I realized this a couple of years ago when I was working on this project that was a 10-plex assay where we had 6 antigen beads and 4 control beads. And at the time, I was doing the standard microgram per million bead coupling. And the purpose of the assay was to detect antibody levels to 6 different pathogen-specific antigens. And the sera we were using were from animals that were infected with different amounts or different combinations of these 6 different pathogens. So the 6 antigens had these different molecular weights, as you see here. Okay, they're not very different, but we were finding that there were some sera that should have been reactive, say, for one of these, and we're picking up still significant signal for some of the other antigens, which we should not have been, because not all these antigens are related enough to be able to should have been that cross-reactive. So what we did was we decided to try, some of the standard different microgram coding levels with a couple of picogram per million bead coding levels and see if we could refine the assay sensitivity a bit and specificity. So generally, when you start doing these kinds of couplings, we recommend that you test whatever proteins you're coupling at a couple of different levels and figure out which one works best for your chemistry and for your assay. So we had sort of a 5 microgram, 2.5, and 1.25 microgram per million beads. And there was one serum that should have been specific for the highest molecular weight protein, and not some of the smaller ones. And as you can see in these microgram per million bead couplings, we're getting a pretty good signal here. And the values we have over here is not MFI values. This is sort of, the ratio to IgG strip serum, okay, because we're measuring the IgG reactivity to these antigens in these serum samples. So this is actually a ratio to background, or a signal to noise ratio, you might say. And you can see that some of these things here are not that high. Darn it, I'll step away from the microphone. I can't take it with me, but that's okay. But they're only going up to a few hundred here. This one may be up to about 1,000 or so, at these microgram per million coding levels. But some of these are still pretty high, and the other ones that we don't want to detect. So we decided to try coupling these at different sort of picomole per million beads. And what you can see here is at a 20 picomole, a 10 picomole, and 1 picomole, you can see that the fold difference over background is much higher. We're now up into the thousands for this serum-specific response. And we still have some high responders down here for the other ones, but once we get down to, say, 1 picomole per million beads, this has almost one of the highest signal-to-noise ratios. And the signal on the non-specific ones is really dampened down with the 1 picomole coating. Okay, because in this chemistry here, theoretically, we have the same number of molecules per bead. So for example, 20 million, the 20 picomole is what probably I think on the order of about 12 million molecules per bead, something like that. But we have ways of determining that, if you want to know how many molecules you actually are coupling to the beads with the molar couplings. So some of the other advantages to doing these molar couplings is that, you can use less protein. So, for these microgram per million beads, you're going to find out that you're probably using a lot more protein, which can be more expensive. But if you start coupling at these molar values, so for example, in this particular assay, if you're coupling at 5 micrograms, 2.5, or 1.25, for every million beads, that's how much protein you need. Whereas if you're going to couple these different antigens, say at these different picomole levels, you're actually going to end up using these amounts of micrograms of protein per million beads, which is significantly less than the 5, 2.5, and 1.25 per million beads. So you're going to save yourself probably a lot more money with regards to how little protein you need. You can boost your sensitivity and specificity, which is certainly an advantage there. And in this case, for this assay, we were doing equimolar coupling. So this equimolar is good for some serological assays like this. Or again, if you're screening antibody clones for people that are generating recombinant antibodies and they want to determine how specific is it for this protein versus another, you can use this equimolar coupling to kind of refine that kind of sensitivity and specificity in your assay. But equimolar coupling is not always good for every application. And one example of that is this assay we built with the University of Rochester. And Sherry talked about this a little bit in her talk this morning, where we were coupling the extracellular domain of spike, which had a molecular weight of 130 kDa. And we were coupling the receptor binding domain of spike, which is about 4.5 times smaller than spike. And we're also coupling nuclear capsid, which is 46,000 and about almost 3 times smaller than spike, and for which I've had a hard time finding a full 3D structure. So I had to pop the sequence in here. I can find the N-terminus and the C-terminus, but not the whole thing right now. But anyway, so what we did when we developed the assay was we coupled, it was a 5-plex assay where 3 of the beads were specific for the spike, RBD, or nucleocapsid. And we coupled the spike at 10 picomoles per million beads and the 2 smaller proteins at 100 picomole per million beads. And then the control beads we had in there was an IgG and an instrument control bead. So the reason why we did this was because they wanted both the spike, the RBD, and the nucleocapsid to give them all positive signals whenever they had a COVID-19 infection that they were looking at. Because if you didn't, adjust the coating level on the beads, then, you're not at certain stages of an infection, all 3 of them won't be positive. So this is from the first paper where we were looking at it in the FM3D, and we later, as Sherry was mentioning, brought this over to the iFlex, but we did also neutralization in both instruments. But the thing here was, this is when we were just comparing how well the assay performed with these 3 different coding levels compared to the Abbott Architect system with all PCR positive COVID samples. And what we could see is that, of course, in the early days of infection, we didn't really get all 3 of them to coordinate with, giving a positive call, but as we got further out, at about 11 days or later in infection, we could then see that because of this, coating level to compensate for the sizes of the different proteins, because remember, spike is a lot bigger, there's a lot more epitope binding sites on it than there are for the nucleocapsid or the RBD. So by doing that shift in the coating level, not making them equal, we could, have them all correlate to, a positive call as we got later in infection. And overall, it worked a bit better than the Abbott Architect system, which was the standard at the time. So in order to do these microgram and molar couplings, we developed some tools to help you do the calculations because they can be kind of complicated. And the calculators available are all Excel spreadsheets. And we have one for doing the microgram coupling of micrograms of protein or peptide per million beads at several different scales. And I didn't talk about the avid-encoded beads, but there's some workbooks that if you want to do something similar with the avid-encoded beads, we have a workbook for that as well. And then if you're going to do your molar coupling, then we have a workbook that can do that at different scales, as well as for binding molar amounts of the avidin beads. And the workbooks are, some of them can be easy to use, but they're a little overwhelming at first. But generally, when we train people on how to use these, once they get the workflow, they see the versatility in using this to get through their couplings and do them accurately, repeatably, and make sure that the stoichiometric ratios, you can keep them better if you do these couplings this way, where you're doing everything, say, per million beads. So this is the one for doing the microgram coupling. And then this is one for doing the avidin binding at micrograms per million beads. And then if you want to do the molar coupling, there's this one here for doing the molar coupling. And this one's a little more complicated because you have to enter in, the proteins you're using, what their molecular weights are. And then because they have different molecular weights, you're going to need to calculate how to make your stocks. So when you're going to do the coupling of however many different proteins of different molecular weights, you can get the concentration of that stock to the right concentration you're going to need for doing your molar couplings. And on this workbook here, it'll also tell you, depending on the molar coating level you've picked, essentially, how many molecules per million beads or per bead you're going to get when you do the couplings. And then there's a tool for doing that with the avidin binding as well. Now, while getting your coupling done correctly, say with these workbooks or some other method, is critical, the other critical thing about building your assay is making your bead mixes correctly. So as one of the other presenters was talking about earlier today, after they do their coupling, they resuspend their beads at 2 million per ml, which is a good concentration if you're, if you're coupling more than 2 million in a coupling reaction, because you're generally not always going to get that amount of beads back. Sometimes there's bead loss after you've done your coupling. And so there's a couple of workbooks to help you track that as well, okay, that we have also. And this workbook has 2 different tabs in it, because what you'll do after you do your coupling is you can use a hemocytometer and a microscope. There's a couple of digital counters you can use to count the bead concentration. And then you would enter it on this information here where the green fields are, and I didn't want to go forward, I just needed the laser, and that's a little too far back. Yeah, let's see here. Okay, so these will help you, you get your bead concentration, and then the value that goes in this cell here, or come on here, yeah, and then there's a cell here where you can say, I want to get all my beads to the same concentration, no matter what they are, because once you've got them all to the same concentration, then that makes making the bead mix a lot easier, where you can then use this other bead mix calculator so that no matter what plex you're using, and they're all at the same concentration, and generally we recommend 2,500 beads per ml,